Welcome to our sixth video on molecular biology. In this video, we are going to be looking at the structure of DNA and RNA. Okay, so when we think of DNA and RNA and we link it to the unit that we've been looking at these biological molecules, and we're looking at different polymers of molecules, okay? And in this case, we're looking at polymers of nucleotides. So if we think of the structure of DNA, we've got these individual monomers called nucleotides, and here we've got an example of a nucleotide. Okay, so we've got a phosphate group, we've got the sugar group, in this case deoxyribose, and we have what we call our nitrogenous base. Okay, and in terms of our bases, here we've got adenine, thymine, guanine, and cytosine. So those, the letters, that's what that stands for. And you see we've got our sugar here is deoxyribose. So this is a nucleotide that will make up DNA. Okay, and this will become more clear as we keep talking. So DNA is this long molecule made up of the monomers called nucleotides. So our carbohydrates, the monomer was saccharides or sugars. Our proteins, the monomers are our amino acids. Okay, and in this case we're looking at DNA, so our monomers are our nucleotides. Now each nucleotide, as I said, has a sugar. We're looking at DNA, so it's deoxyribose. If we was looking at RNA, it would just be ribose. Each nucleotide has a phosphate group, that's the same in both DNA and RNA. And each nucleotide has a nitrogenous base, so a base. Okay, that's the same in DNA and RNA. The bases differ between the two. Okay, so in DNA, there's adenine, thymine, cytosine, and guanine. They're our four bases. So over here, this can be one of four bases. So the sugar and the phosphate part are the same in all four nucleotides. So whether they've got adenine, thymine, cytosine, or guanine, the sugar and the phosphate group is the same. Okay, it's the base that it contains is different. Now, when we look at the structure of RNA, okay, RNA again is a polymer of nucleotides. Each nucleotide has got a sugar again, but in this case it's ribose sugar. It's got a phosphate group, and they RNA monomers, so nucleotides have nitrogenous bases, the same as DNA. Now we've got four bases here. We've got adenine. Instead of thymine, we've got uracil, cytosine, and guanine. Okay, the sugar phosphate part are the same in all of the nucleotides, but just like in our DNA, it's the bases that make them different. Alrighty, so looking at nucleotides. In DNA, we said, so looking at the bases in the nucleotides. In DNA, we said we've got the four adenine, guanine, cytosine, and thymine, whereas in our RNA, we've got uracil, which replaces thymine. Okay, so your understanding one is you need to know that DNA and RNA are made up of these polymers of nucleotides. Okay. Alrighty, so how do we form our polymer chain that we find in DNA? So to form a DNA molecule, the nucleotides are linked together. So if you see here, you've got one nucleotide, you've got another nucleotide, and you've got another nucleotide and another nucleotide. Now they are bonded with this strong covalent bond here. So the phosphate group of one nucleotide is linked with a covalent bond to the deoxyribose sugar in the next nucleotide. Remembering we're talking about DNA here. So this forms what we call the backbone of the DNA molecule. Okay, so looking at this diagram here, phosphate group, deoxyribose sugar, and then we've got our base. So this base is adenine, okay? So here's another molecule, here's another molecule. Now you'll see this bond here, sorry, this bond here between the phosphate group and the deoxyribose sugar, okay? Those covalent bonds along there. Okay, so in DNA, basically what happens is we've got that phosphate backbone, so we call it, okay, where our nucleotide bases are all joined together. So along here, our phosphate backbone. Then we've got our deoxyribose and our base. Now what happens through base pairing, which you would have heard about in year 10, okay, two DNA strands actually bond together as such. Now the base pairing, so you've got thymine and adenine. So remember, apples fall from trees and cars go into garages, okay? And guanine and cytosine. So they're our base pairs. So adenine will only pair with thymine, guanine only pairs with cytosine. And what happens is we get these hydrogen bonds between the bases. And that causes those rungs on the ladder that form with DNA. So we've got two backbones, and then they're joined together by the bases and that base pairing. So two strands of nucleotides are linked together by the hydrogen bonds with our pairs. So remember, apples come from trees. So adenine pairs with thymine, cytosine pairs with guanine, cars park in the garage. Okay, the two DNA strands run opposite each other. We call them anti-parallel because yes, they're parallel, but one of them's flipped, so that if they were parallel, then the bases would be, for example, both on the right hand side or both on the left hand side, but they're not. One set of bases is on the left, one's on the right, so that they can join. So that's what anti-parallel means. This will then form the double helix as it then twists into shape. And so we say our purines always pair with the pyrimidines, okay? So we're just talking about the end of the adenine, the end of the base. Sorry, muddled my words there. Thymine, cytosine, and uracil are examples of pyrimines, while adenine and guanine are clarified as purines. Okay, so keeping on thinking of that forming DNA, and if we look at this diagram here, okay, we can see our hydrogen bonds. We've got our backbones of our DNAs here. Okay, so we've got the side of the ladder and the rungs of the ladder. Don't call it that in exam conditions, but that's something you can remember it by. When many nucleotides join together, they form this chain. A bond forms between the sugar of one nucleotide and the phosphate of the other, so we get the backbone forming. We've already spoken about that. One chain runs from head to tail with the phosphate at the head, so that's here. Okay, this is also known as the 5', okay, 5' end. The opposite has the sugar molecule at the head or the phosphate at the tail. So we've got the phosphates are your 5' end, whereas your sugar molecule is the 3' end. Okay. Alrighty, having a look at the difference between our DNA and our RNA nucleotides. Okay, ribose is pentose sugar, as is deoxyribose, so pentose meaning 5. Okay, so 5 carbons. So the difference between ribose and deoxyribose, deoxy, okay, our ribose has the hydroxide here, whereas deoxyribose doesn't have that oxygen, so it's just got the hydrogen there. Other than that, the difference comes in the bases, where RNA has got uracil and DNA has the thymine. So they're the differences between those nucleotides. Again, just looking at the difference in those sugar molecules. Okay, ribose has the hydroxide, deoxyribose doesn't have that hydroxide there. And that is the same slide. Okay, so again, this is a good graphical representation of the differences between DNA and RNA. Now, we've just looked at how a DNA molecule forms, okay? Now, RNA is exactly the same. The way that the sugar phosphates bond to make the backbone of the ribose is, the backbone of the RNA is exactly the same. The only difference is in the RNA, the bases don't at this point bond with another base, okay? RNA is a single chain, DNA is a double chain, okay? So that double helix we call DNA, whereas RNA is a single chain. Half a ladder, basically. Both of them share the bases cytosine, guanine, and adenine. Uracil is a unique base to RNA, whereas thymine is unique to DNA. The other difference being the sugars that we just spoke about. Okay, this is in table form. Okay, so if we're comparing DNA to RNA, the number of strands in a molecule in DNA is 2, and in RNA is 1. The type of sugar in deoxyribose in DNA is deoxyribose, and in RNA is ribose. Those nitrogenous bases, in DNA you've got adenine, cytosine, guanine, and thymine, but in RNA all the same, but you've not got thymine, you've got uracil. Subunits in DNA are nucleotides, subunits in RNA nucleotides, and the nucleotide components are the same in both. You've got sugar base and a phosphate. Now, if you were asked a question of discuss the differences between DNA and RNA and say it was worth 2 marks, you wouldn't be able to say that DNA has 2 strands in its molecule and RNA has 1. That would only be 1 mark. Okay, you'd have to choose 2 of the features and say how they're different to get both marks. You can't just say the opposite. If you was asked how to compare and contrast, comparing is how they're similar. So you'd be saying that DNA and RNA are similar because they're both made up of a sugar, a base, and a phosphate. They're subunits for both are nucleotides. However, they are different because DNA has 2 strands in its molecule, whereas RNA has 1. DNA is the sugar component of DNA is deoxyribose sugar, whereas the sugar component of RNA is ribose. And then you can say they both contain the nitrogenous bases of adenine, cytosine, and guanine. However, DNA contains thymine, whereas RNA contains uracil. So if you're asked to compare and contrast, it's saying how they're similar and how they're different. If you're asked to discuss differences, you'd need to discuss 2 if it was for a 2-mark question. Okay, so your application 1, Crick and Watson's elucidation of the structure of DNA using the model making. So you see there, using in the picture, you see Crick and Watson's initial ideas around what DNA looked like, and they kind of managed to put together the double helix model. So models are used in science as they give a more realistic impression of what a proposed structure is like. So it's really important to be able to visualize things in 3D, and we looked at that in one of the previous videos where you was looking at the computer generated images in 3D. Models are not always 3-dimensional though. They can be a theoretical concept and represent systems or processes. So you could have a diagram that's representing the model of how something works. A common feature of models is that they are proposals, they're ideas, they're saying this, we think this is how it works, and therefore they're there to be tested. Now, model making played a vital part for Crick and Watson's discovery of the structure of DNA, but it took 2 attempts before they were successful. Crick and Watson used pieces of cardboard in the shape of DNA bases to represent their shapes and found that complementary base pairs could be formed. So it was literally, they were literally working on a puzzle and made this discovery. Okay, so that's an overview of the actual understandings that you need from this unit. We are going to be practicing drawing the nucleotides and making sure we get the shape and that all correct, and many other things that go with this unit in class. But this is important, along with 1.5 and 6, which we will review later in this or in the next video before we commence 2.7. So this is the base of looking at transcription, translation and replication that is coming up soon. Hope you enjoyed the video. See you next time.